Welcome back to the Deep Dive.
Great to be back.
And a special welcome if you're joining us for the SANS Science Experience.
Yes, welcome everyone.
This is where we take the most exciting new research, preprints sometimes too, and we really unpack them for you. Today, we're diving into, well, the fascinating world inside our cells, specifically our mitochondria.
The powerhouses.
Exactly, the powerhouses. And there's this really elegant puzzle about them. How do these tiny organelles coordinate making their own energy machinery?
It truly is a marvel of biological engineering when you think about it.
It really is.
Mitochondria have this, well, unique setup, a dual genetic system.
Dual genetic.
Oh, yeah. You can sort of picture it like a company with two different manufacturing arms.
All right.
So the main factory, let's say that's our nuclear DNA that makes most of the parts, ships them in.
Right, imports them.
But then there's also this small on-site workshop. That's the mitochondrial DNA, or mtDNA. And it produces just a few absolutely critical components right there inside the mitochondrion, essential for energy production.
So you've got parts being made in two totally different places in the cell.
Exactly.
And they somehow have to come together perfectly to build this oxidative phosphorylation system, OXPHOS.
That's the one, OXPHOS. It's the machinery that generates, the vast majority of our ATP.
So our energy currency.
The cell's energy currency, precisely. And it's not just about getting the pieces together. The assembly has to be incredibly precise. If it goes wrong, you risk cellular damage. Things like proteotoxic stress.
Which is like a buildup of junk proteins, right? Misfolded stuff.
Exactly. Misfolded proteins gumming up the works. And also the production of harmful reactive oxygen species, ROS.
Nasty molecule.
Very nasty. They can damage all sorts of cellular components.
Okay, so that coordination is key.
And that's really the heart of our deep dive today. We're exploring how the translation, that's the protein-making process from the mitochondrial DNA is specifically tailored, you could say, to the assembly of these XPHOS complexes.
How it's all synced up.
Exactly. It's a story full of, well, intricate feedback loops, specialized machinery, all working together.
Making sure everything clicks into place at the right time in the right amounts.
Precisely.
Okay, let's unpack this. This mitochondrial translation system. It's pretty unique, isn't it? Different from the rest of the cell.
Oh, definitely. You have to remember their origins.
The bacterial connection.
Exactly. The endosymbiotic theory, mitochondria, likely evolved from bacteria engulfed by early cells. So they've kept some unique features in how they express their genes.
Like what, for instance?
Well, their messenger RNAs, the blueprints, they lack things called 5 caps and Shine-Dalgarno sequences.
Which are important elsewhere.
Crucial. In our main cellular machinery, and the bacteria too, they help the ribosomes, the protein factories, find the right starting point on the mRNA.
So how do mitochondrial ribosomes even know where to start?
That's actually a really fascinating area still being researched actively.
It's different. And the ribosomes themselves, they're not standard issue either.
Not at all. Mitochondrial ribosomes are remarkably protein-rich compared to, say, bacterial ones or the ones in our cytoplasm.
More proteins.
Yeah. In humans, the mitochondrial ribosome, it's called the 55S ribosome, has over 80 proteins. Many are unique. No direct equivalents in bacteria. Highly specialized.
Specialized for the mitochondrial environment.
You got it. And their location is strategic, too.
They're not just floating around.
Nope. They are tightly associated with the inner mitochondrial membrane.
Which is where the RXPHOS complexes end up.
Exactly. And this closeness, it helps facilitate something called co-translational insertion.
Co-translational, meaning as they're being made.
Precisely. The proteins made from mtDNA tend to be very hydrophobic.
Water-hating.
Right. So being made right at the membrane means they can immediately slot themselves into it as they emerge from the ribosome.
Avoids them clumping up in the watery interior.
Exactly. Prevents aggregation, potential problems, and there are helper proteins involved too, like MB1 or ML45 in humans, and an insertase called OXA1 or OXA1L in humans.
So they help anchor and insert these proteins.
Kind of like a specialized construction crew, building parts directly into the structure as they're manufactured.
That makes a lot of sense logistically. Build it where you need it. Okay, so the mtDNA parts are made on site, but assembling the whole OXPHOS complex, that sounds like a huge coordination challenge, bringing in all the nuclear-encoded parts, too.
It is an incredibly coordinated effort. You need the precise interaction of both the nuclear and the mtDNA-encoded subunits.
And it doesn't just happen spontaneously.
Oh, no. There's a large, dedicated team of assembly factors involved. Absolutely essential.
Assembly factors. What exactly are they doing? They sound crucial.
They really are the unsung heroes. They act as molecular guides, helpers.
Like chaperones?
Chaperones, yes, but also more. They help with folding the subunits correctly. They ensure crucial cofactors like metal ions get added at exactly the right time. They stabilize partially built structures, shepherd the subunits into the growing complex. Without them, it'd be chaos. Or just non-functional junk.
Can you give us an example? Make it a bit more concrete.
Sure, let's take complex 4. Mammalian complex 4, also called cytochrome C oxidase, vital enzyme.
Right, in the electron transport chain.
That's the one. It has 14 different protein subunits, but guess how many are made by the mtDNA?
Maybe half.
Only 3. COX1, COX2, and COX3.
Just 3 out of 14. Wow.
Yep. The other 11 are made in the nucleus, imported. But here's the kicker. Assembling just this one complex involves more than 30 different assembly factors.
30 for one complex.
30. And they're critical for things like inserting Ham-and copper ions into COX1 and COX2.
The metal bits.
Essential cofactors for handling electrons safely. Getting them in right prevents damaging free radicals.
Okay, that's incredibly intricate. How does the cell manage the order? It can't just be throwing everything together.
No, it's very orchestrated. A modular assembly pathway.
Modular, like building blocks.
Exactly. For complex 4, it starts with a module built around the COX1 subunit. There's a protein complex called MITRT that helps with that.
Okay, step 1, COX1 module.
And then that module hooks up with another one containing COX4 and COX5A. Then COX2 gets integrated, then COX3.
Sequence is critical.
Absolutely. And finally, a subunit called NDUFA4 joins to complete the functional enzyme. It's like specialized Lego bricks clicking together in a specific order.
A molecular dance, really precisely choreographed. Now, I know a lot of the early understanding of this came from studying yeast, right?
Absolutely correct.
Yeah.
Budding yeast, Saccharomyces cerevisiae, has been just invaluable as a model system for figuring out these fundamental processes.
Simpler system, maybe?
In some ways. Its mitochondrial DNA is smaller than ours, about 86 kilobases. It encodes 8 proteins total, including parts of complexes 3, 4, and V, plus a ribosomal protein.
But not complex I or 2.
Interestingly, no. Yeast mtDNA doesn't encode any subunits for those 2 complexes, unlike ours.
And their mitochondrial mRNAs, they're also structured differently from mammals.
Yes, quite different. Yeast mito-mRNAs have these long sequences at the beginning, the 5-foot untranslated regions, or 5 UTRs.
Untranslated, but regulatory.
Exactly. They don't code for protein, but they control things. And unlike most eukaryotic mRNAs, including ours in the mitochondria, yeast mito-mRNAs lack poly-A tails at the end.
Okay, so long 5-foot UTRs, no poly-A tails.
And those long 5-foot UTRs were a big clue. Researchers found proteins, called translational activators, that bind specifically to these UTRs.
And they control the translation, like switches.
Pretty much.
Yeah.
Like specific on-off switches or maybe volume knobs for making different mitochondrial proteins.
Fascinating.
And what's really cool is the discovery of direct feedback loops in yeast.
Feedback loops. You mean the assembly process talks back to the production line?
That's exactly it. The assembly status of a complex actually influences how fast its own mtDNA-encoded subunits get translated. Beautiful self-regulation.
Okay, now this sounds really clever. Tell us more about these loops in yeast.
Okay, take cytochrome B, a key part of complex 3. Its translation is directly linked to complex 3 assembly via a protein complex called CBP3-CBP6.
So if assembly stalls, translation slows down.
Something like that, yes. And for complex 4, there's a similar story with COX1, the central subunit. Its translation is tied to assembly through a protein called MESS51.
MESS51, how does that one work? Does it do double duty?
It does. It's really sophisticated.
Yeah.
MESS51 acts as a translational activator, binding the COX1 mRNA's 5-foot UTR to boost translation.
Okay, turns it up.
But it also interacts with the brand new COX1 protein during the early steps of complex 4 assembly.
Ah, so it gets pulled away.
Exactly. As MESS51 gets tied up in these assembly intermediates, it's temporarily unavailable to activate more translation.
So it naturally slows down COX1 production if assembly isn't keeping up.
Precisely. Only when assembly moves forward and MESS51 is released can it go back and activate more COX1 translation. It's a self-adjusting mechanism. Prevents wasteful overproduction.
That is genuinely ingenious, making sure you don't build parts you can't use right away.
Didn't I read somewhere that heme is involved with MESS51 too?
Yes, that's a crucial layer. MESS51 binds heme, the iron-containing cofactor essential for complex 4 FES function.
Right.
And binding heme is critical for MESS51 to work properly as a translational activator.
So it links cofactor availability directly to subunit production.
Yeah. It's like the system checks if all the necessary ingredients are there before ramping up production of a key structural part. Amazing coordination.
It really is.
Yeah.
What about complex V, the ATP synthase? Same kind of link?
Absolutely. The translation of FP6P and KAP, two mtDNA-encoded subunits of complex V in yeast, is activated by partially assembled intermediates of the F1 part of the synthase.
So again, makes the parts when the rest of the structure is ready for them.
Prevents building up isolated components.
And it's not just these specific activators you mentioned, even ribosomal proteins can play a role. That's right. There's evidence, for instance, that a mitochondrial ribosomal protein called ML38 influences COX-1 translation, and this seems sensitive to how well complex 5E assembly is proceeding. It's a whole interconnected web.
A web of regulation.
And you really see the importance of this when yeast cells shift how they produce energy, say changing their food source. This regulation is key to that adaptation.
Okay, so yeast gave us this fantastic blueprint for understanding coordination, but mammals, our mtDNA is different. Our mRNAs are different. How does it work for us?
It is different. Yeah, mammalian mtDNA is much smaller. Our mRNAs have very short or sometimes no 5' UTRs.
So that yeast mechanism with activators binding long 5' UTRs, probably not the main way.
Less likely to be the dominant strategy, yes.
Yeah.
And we generally don't find clear human equivalents, functional homologs of those specific yeast translational activators. Plus, our mitochondrial mRNAs do have poly A tails.
Okay, so if we don't mostly use those 5-UTR activators, how do mammals coordinate mitochondrial translation and OXPHOS assembly? Is the picture clear yet?
It's still emerging, definitely more complex to study than yeast, but we are identifying key players and feedback mechanisms.
Are there any examples of mammalian translational activators?
There are. One well-studied example is TICO1. It specifically binds the COX-1 mRNA and promotes its translation.
COX-1 again, the complex 4 subunit.
That's right.
And
mutations in TECO1 cause Lee syndrome, a really severe neurological disorder linked to complex 4 deficiency. So TECO1 is clearly crucial.
Okay, so the principle is there, even if the players differ. What about those feedback loops, linking assembly back to translation? Are we finding those in mammals too?
Yes. We're starting to uncover them. It's really exciting. One example also involves complex 4 and COX-1 translation.
Okay.
Components of that miTRK assembly complex we mentioned earlier, specifically proteins called C12RF62 and miTRK12, they bind to the COX-1 protein as it's being made, co-translationally.
Right on the ribosome.
Seems like it. C12RF62 actually interacts directly with mitochondrial ribosome. Now, if early complex 4D assembly gets stuck, maybe miTRK12 is missing or another early subunit,
then what happens?
The ribosome, with the growing COX-1 chain still attached to C12RF62, basically stalls. It gets stuck.
And that stalling sends a signal.
Effectively, yes. That stalled ribosome nascent chain complex leads to a reduction in the overall production of new COX-1 protein. So translation rate is directly linked to assembly progress.
That sounds remarkably similar in principle to the yeast SSFE1 mechanism.
It does, doesn't it? Different molecules, different details, but the same fundamental outcome.
Yeah.
Linking production to demand.
Evolution, finding different ways to solve the same problem. Any other examples? Complex 3, Complex 1.
Yes, evidence is building for complex 3 as well.
Yeah.
There's an assembly factor called UQCC2. Mutations cause complex 3 deficiency.
Right.
And also a decrease in newly made cytochrome B, the mtDNA-encoded subunit. It seems UQCC2 stabilizes another factor, UQCC1, which binds the nascent cytochrome B. So a link there too. And for complex I, research is pointing towards a possible link between its assembly and the translation of ND2, one of its mtDNA subunits. A protein called miTRK15 or COA1 might be involved, maybe associating co-translationally and promoting translation.
So these feedback loops seem to be a conserved strategy, even if the details vary.
It really looks that way. It highlights just how fundamental this tight coordination is for keeping our cells healthy and powered up.
It's amazing. And you said our understanding in mammals is catching up. What's driving that progress now? New tools?
Absolutely. 2 recent breakthroughs are really transforming the field.
Oh, yeah. What are they?
First, the development of DDA-derived cytosine base editors.
Base editors for mitochondria.
Exactly. They let us make precise, targeted changes to the mitochondrial DNA itself. That was incredibly difficult, almost impossible until very recently.
Wow. Okay. That's huge. What's the second one?
The use of morpholinos. These are synthetic molecules like custom-designed blockers.
Blockers for what?
You can design them to specifically block the translation of individual mitochondrial mRNAs.
So you can selectively turn down the production of just one protein.
Precisely. So between the base editors to change the mtDNA blueprint and morpholinos to block specific translation, we now have much finer tools to probe these networks.
Tools to really tinker with the system and see what happens.
Exactly. They are game changers. They're going to let us unravel these coordination mechanisms in mammals in unprecedented detail.
That's incredibly exciting for the field.
It really is. We're poised for a much deeper, more comprehensive understanding of how these powerhouses are built and regulated.
Well, this has been a truly illuminating deep dive. The sheer elegance of how our cells manage this dual genetic control, making sure these energy factories are assembled just right. It's remarkable.
It really underscores the beauty and, frankly, the complexity of cell biology. And it's not just academic curiosity, is it?
No, definitely not.
Understanding this is crucial for getting grips on mitochondrial diseases. Many are caused by defects in these very processes.
Right, which can have devastating health impacts. It has real clinical relevance.
Absolutely. Potential for better diagnosis, maybe even future therapies down the line.
It really makes you wonder, doesn't it? This level of intricate coordination, feedback loops, localized production, are similar principles likely at play in other parts of the cell, too, for other complex structures?
That is a fantastic question. And yes, researchers are actively looking into that. These ideas, localized translation, assembly factors, feedback control, they might well be fundamental strategies used across many areas of cellular organization. A lot more to discover there.
Definitely.
Well, a brilliant thought to leave our listeners with. Thank you so much for walking us through this intricate world today.
My pleasure. Always fascinating to talk about.
And thank you all for joining us for this deep dive, part of the SANS Science Experience. We hope you found this exploration of mitochondrial coordination as fascinating as we did.
Hope so.
If you enjoyed it and you want to see more deep dives into cutting-edge science, please do like this video and subscribe to the channel. It really does help support what we do here.
It does indeed. Thanks again for tuning in, and we'll see you next time.